My name is Zach Wilson, and I am presenting on how circadian rhythm is thought to accommodate requirements of fetal development during pregnancy. So, circadian rhythm is a cyclic fluctuation in hormonal release of an organism. The needs of the body change throughout the day, the week, the month, and we know that pregnancy alters this normal homeostasis. This is because fetal development is energy intensive, and normal metabolic regulation must change to accommodate this. So the article that I'm presenting on today is titled Pregnancy-Induced Changes in the Circadian Expression of Hepatic Clock Genes, Implications for Material Glucose Homeostasis. So the design of this experiment revolved obviously around female rats because they were the ones impregnated. They were fed ad libitum and housed under two different conditions. The first being a normal condition of 12-hour light, 12-hour dark, and the lights on at 7 a.m. The second condition was a reverse 12-hour light, 12-hour dark cycle with the lights on at 7 p.m. This is just a control mechanism, and we saw no significant difference in either groups. So the first protein that we looked at was called BMAL1. And BMAL1 binds to a protein called clock, and this forms a heterodimer. This heterodimer binds to a downstream promoter and activates the transcription of further clock genes involved in circadian rhythm. So we see here in this graph, as we go from non-pregnant to day 6, to day 10, to day 14, to day 18, we see that the amplitude of this curve is being decreased. And this is a p-value of 0.05. Here we see the counterpart of the BMAL1 protein, and this is clock. So the levels of clock correlate to BMAL1, and we see here from non-pregnant to day 6 to day 10 to day 14 to day 18, we see a decrease in the amplitude. And this is showing the change in the fluctuation or the change from circadian rhythm to a more consistent expression of this gene. And this is due to the fact that because the energy needs of the female rat are evolving or are changing away from the normal due to the increased glucose requirement of that fetus, we see that these hormones involved in glucose regulation are turning towards more consistent expression. So the protein we looked at here is called pair1, and pair1 is a downstream target of the BMAL1 clock heterodimer. This is involved in a negative feedback mechanism. And this protein is involved in more regulation of downstream protein synthesis, proteins involved in circadian rhythm. And here we see, again, from non-pregnant to day 6 to day 10 to day 14 to day 18, we see a decrease in the amplitude away from the normal circadian rhythm to a more consistent expression of this protein involved in glucose regulation in the body, with a p-value of 0.05, statistically significant. Here is just another pair protein. This is pair 2, and again, it's a downstream target of the BMAL1 and clock heterodimer complex. Again, from non-pregnant to day 18, we see a decrease in amplitude away from the normal circadian rhythm to a consistent gene expression with a p-value of 0.05. So this shows the oscillatory expression of two of those genes involved in the negative feedback mechanism. We see here the BMAL1 and the pair1 expression, just a decrease in amplitude towards consistent expression. So the protein we're looking at here is called GLUT2, and GLUT2 codes for a glucose transporter. And we see here, starting from non-pregnant to day 6 to day 10 to day 14 to day 18, we see a decrease in amplitude again of this protein away from normal circadian to a consistent expression. So here we see something a little bit different. We're looking at the expression of a protein called GK, and GK is a protein that phosphorylates glucose, and this is essential for in the glucose pathway to ensure that glucose stays inside the cell, and we need this because the requirements of the pregnant mouse is being increased due to the fact that the fetus is requiring more and more energy for growth. So we see here in the non-pregnant day 6 and day 10, we see a decrease in the amplitude. However, we see something different in the next part of that mid-gestation period. We see a turn away from the normal rhythmic pattern, and we see a consistent, statistically significant, consistent expression of this protein. Here we're looking at another protein called PK, and PK is a protein that phosphorylates pyruvate, and pyruvate is a byproduct of glycolysis and can be used in many different biosynthetic pathways. We see here a similar pattern to what we saw in GK, where from non-pregnant to day 6 to day 10, we see a decrease in the amplitude but a maintenance of that rhythm. However, from day 11 to day 18, we see that rhythm is lost away from that, again signaling that the needs for this protein in the glucose pathway is being more and more essential. So in summary, we saw that the regulatory genes of BEL1, clock pair 1 and pair 2, as we saw in figure 2, are the amplitude of these of the expression of these proteins is being decreased, and turning away from that cyclic pattern to a normal consistent expression. And in the glucose metabolism genes, we see that the decrease in amplitude of GLUT2. However, we see the latent loss of rhythm in GK and PK, which we saw in figure 4. So this was the literature cited, and that's it.